Hi, this is Laura Whitman with DNA 2.0, and I'd like to welcome you to our webinar on optimizing gene expression and performance using GeneGPS. Right now, we'll just do a quick bit of housekeeping. If you can hear us and everything's good, that's great. If you can't, you can raise your hand or send us a chat, and we'll see if we can help you out with that. We're also going to do a couple of polls during this webinar, so I'm going to send out the first question for you, and you can answer that. It would be great to have. Your poll window should pop up for you, and you should be able to click on an answer to that. And then we'd like to move on with the show. So I'd like to introduce Dr. Mark Welch. He's the Director of Gene Design here at DNA 2.0, and we're happy to have you.
All right, thanks, Laura. So today I'm going to be talking about how we approach gene optimization at DNA 2.0, very distinct from traditional methods and quite different from the approach other gene synthesis companies use. First, a little bit about our company. We're based in Menlo Park, California. We've been around for about 9 years. We're fully employee-owned, privately held, and what we do is use our expertise in gene synthesis and bioinformatics to provide bioengineering solutions for our customers. So our key technologies to provide these solutions are our gene engineering platform, which we call GeneGPS, and our protein engineering platform we call Protein GPS. In today's webinar, I'll focus on our GeneGPS approach. We'll discuss protein engineering in future webinars, but in many respects, today's webinar can be considered a preview of that, in that both gene and protein GPS are fundamentally similar approaches. Now, the key to both our engineering platforms is what we call infologs. So homologs, we all know what homologs are, naturally occurring genes, share common ancestry and traits. Infologs are synthetic genes that are designed to interrogate the sequence-function relationship. So they're genes that we make to basically ask certain questions about how a sequence relates to function. And I'll give you examples of how we apply this infolog approach in gene engineering throughout this talk, so you'll have a clear idea of really what I'm talking about there. So whether we're talking about gene engineering or protein engineering, our approach is basically the same. So we start with any existing data. Maybe we have no real data. We just have some suspicion of certain variables that may be important. We use that information to choose the sequence variables we want to explore. We design a library of genes that systematically explores those variables, and those are the infologs. We then synthesize those genes, test the genes in a commercially relevant assay, and this is a key to both our approaches. Pull up a highlighter here so you know where I am. Is that because we, and I'll explain this throughout the talk, but because we use infologs and because we can efficiently sample gene variation space, we can reduce the number of tests that we need to do, and that can be very important for certain applications where high throughput screens are not really adequate to mimic performance in real world applications. I'll come back to that throughout the talk. So once we have data in the relevant assay, we can model that data, basically model the sequence function relationship, and we can do this iteratively. Once we've learned something about that relationship, we can choose new variables to explore, or we can explore the most important variables more deeply to come to a more optimal solution. All right, so how do we apply this to gene engineering? Now, the challenge of gene engineering comes from the redundancy of the genetic code for the most part. And for the purposes of the talk, by the way, when we say gene engineering, we're mostly referring to synonymous codon change within the open reading frame of a gene. But keep in mind that the principles of engineering that we're talking about can be applied to any variable in the sequence of the promoter or RBS, for example, in addition to sequence changes in the open reading frame. But for all the examples that I'll describe will be open reading frame substitutions that preserve the amino acid sequence of the protein, just change synonymous codon usage. Um, now, because of the redundancy of the genetic code, there's an astronomical number of ways that you can code for a gene of average size. So there's no possible way we can sample all of this sequence space. The only way we can hope to reliably improve genes is if we have some rules to limit the number of variables. We can reduce the variables to some manageable number that are predictive of expression. But the problem is we still don't have a full understanding of the relationship between gene features and expression. And we know it's complex, but we still don't have it reduced down to the point where we can reliably predict which genes will express well and which won't. And not just that, but which genes would be most optimally expressed. And the problem with real-world applications is that the definition of optimal is not so clear. We don't necessarily want maximal translation rate for optimal production of soluble or active protein in some cases, or maximal production or transport of a protein in the membrane or secreted state. So we want technologies that don't just maximize the rate of protein production from a gene, but we want to produce what the customer cares about, active membrane protein, what have you. We want a method that can basically accommodate the various quirks of different proteins in different production systems. All right, so traditionally what has been done is that people have basically made best guesses about what's important, usually derived from what has been seen in natural gene sequences. And there are a number of different principles that exist in the literature based on these natural variations and best guesses. The problem is that none of them have been thoroughly validated as beneficial, and certainly none of them been shown to be truly optimal, and especially for real-world heterologous expression applications. And also many of the principles that exist are mutually exclusive. For example, you can't control GC percentage completely independently of codon usage bias or mRNA structure. So if all these things are important in some way, you have to figure out some optimal compromise. And if we don't have good rules for that, how do we know we're really optimizing the gene? So our approach is different. Instead of guessing at what's important, what we do is interrogate the cell to figure out what's important. So a couple years back, we published some work in E. coli where we studied expression of two genes, a single-chain antibody and a polymerase for expression in E. coli. And basically what we did was we created a set of genes, an infilog set, and the key to the approach that we used is we used design of experiments to vary, methods to vary the variables of the genes maximally while minimizing the covariation between those variables. And I'll get into details about what variables we look at when we do this. But keep in mind that we're using design of experiments methods to minimize covariation between variables, maximize the search space that we're interested in. We use then gene synthesis to make those genes, test them for expression in E. coli, build a model, and that model then can feed back on what variables are important. The key thing is that we're using design of experiments and gene synthesis combined to sample any variation that is of interest. We can sample any range of gene design variables, and we can sample them efficiently because we're making each of these genes discretely. Now this matrix gives you one example of how we apply this infilog concept to gene design. This is one particular set where we're sampling codon usage frequencies across a gene. And what's shown here, the columns here are individual genes in the set. In this set we have 47 genes. The rows along the column here each represent codon biases for each of the amino acids. And for each amino acid we're varying codon bias in the direction of C content or purine versus pyrimidine content, so a generic variation on codon bias. And for each amino acid, we're varying the codon bias independently of the next amino acid. So across the set, we're systematically sampling codon biases for each amino acid, and we're not, and codon biases for each amino acid are not covariating. So we can independently assess the impact of codon bias for each amino acid. Now, one of the advantages of using infologs is that, and using this particular approach, is that we have full control over the sequence space in our library. So, for example, I'm just showing two types of sequence space control that we have that's advantageous. So in the left panels here, I'm comparing the sequence diversity in a library that was supposedly made by oligo randomization. So they would expect just full randomization throughout the set. And we're comparing that to the randomization on the sequence level in our infilog set. And you can see a lot of clustering in the sequence tree here for a set. This particular set is a set of GFP genes published by Codon in Science a couple of years back. You can see a lot of clustering indicating that their randomization wasn't truly random. This is something we see commonly when we use oligo-based randomization methods. There's always some bias in the assembly, things that might affect which oligos get incorporated more than others. But since we're making our genes in silico, essentially, we're designing them to be different from each other, and we make exactly what we design, we can make sets that show no covariation gene to gene, so no clustering, so much more broad sampling of sequence diversity. Now the right panels show a different type of diversity that's important to us, covariation between codon usage frequencies. So each of the columns and rows in these matrices are individual codons, and the intersection between those indicates the degree of covariation between those pairs of codons, whether it's positive or negative. You can see in the random set, just because of the limitations of the oligo-based approach, that there's a lot of covariation between codons. Basically, any codon is highly correlated positively or negatively to every other codon. So you get deep color, blue or red, throughout the matrix. But in the infilog set, there's minimal covariation between different codons. So they're not. So when we see an effect, say, for a particular codon that it correlates with expression, we know it's that codon. It's not something else that may be covariate with it. We do see a few dark blue spots, but those are necessary anti-correlated codons where the amino acid only has two choices. So we really have minimal covariation between both codon usage as well as minimal covariation in local sequence diversity in these sets. All right, so what do we see when we put an infilog set into E. coli and test for expression? And this is all results that, for the next couple slides, that we published in our PLOS One paper, so I'll refer you to that for this data, more data, and some of the interpretation. We'll just gloss over it in this talk. But the one thing that we saw is that there was no correlation between expression and the codon adaptation index. The codon adaptation index is a measure of the relative use of codons that are more highly used in genes that are highly expressed in the host. And this is the principle, the main principle for design for most codon optimization algorithms that exist today, and most companies use this as their main principle of design. So they're trying to maximize the codon adaptation index. A codon adaptation index of 1 means you're only using the so-called optimal codon for every amino acid in the sequence, and that's the one that is most frequently used in highly expressed genes. So everyone tries to push codon adaptation usually up above 0.9 in our optimization algorithms. But we see no justification for using this as a principle of design in our infilog sets for both proteins, the polymerase and the single-chain antibody. We see no correlation with the codon adaptation index. In fact, our highest expressing genes have more intermediate codon adaptation index levels, and we see fairly poor expression for most of our high codon adaptation index genes. And we also did not see a strong correlation for these gene sets between RNA structure energy near the initiation site of these genes. And that's in contrast to an earlier study by Kudla, the same one I referred to in the previous slide, where they did see a strong correlation with RNA structure. I should mention that they also did not see a correlation with the codon adaptation index, and a number of other studies recently out in the last couple of years have shown the same thing. So we're not alone in not seeing a correlation, a strong impact of this index on heterologous expression. But again, other studies, Kudla and others, have shown that RNA structure is important. We're not seeing that in this set, and that just turns out to be a quirk of the particular proteins that we studied. The propensity to form RNA structure is highly dependent on the amino acid sequence of the N-terminus of the protein, and it just turns out that our genes didn't sample strong structures. In the Kudla study, it was structures that were stronger than -9 that showed a strong effect. We just aren't sampling in that range. So we don't see, so all we can really conclude is that there's no correlation in the range from, say, -7 or weaker. We do see clearly that there's something more going on than just the 5' end sequence, irrespective of structure. So the red arrow here indicates a number of genes that have identical codon usage for the first 15 codons or more in these genes. And we see their range of expression can span over the entire range of expression in the data set. So there's a lot more going on that controls expression than just that coding sequence at the beginning of a gene. So if it's not these things, what is responsible for the variation we see? So the way we approach this, and we have a number of different methods, and we have our favorites, but usually we apply what's called multivariate regression. We input the entire matrix of variables and the expression data that we get. And we use regression to basically search for correlation with the variables and expression. And the regression finds optimal weights for each variable that best explain the expression variation that we see. And then we use an array of statistical tests to determine whether it's significant, and among the results of the model, which are more significant than others. And I'll show you some examples of that. You should keep in mind that this doesn't have to be just the variables that we use to design the gene. We can input any variable in the gene into the regression, things that may not have been part of the design of experiments method to begin with. So just input everything, see what works. So what we get when we look at these two genes in E. coli is that not only can we fit models to each of the data sets for the polymerase and the single-chain antibody, but a single regression can explain most of the variation we see for both sets simultaneously, and that's shown here. So in this slide is a model using a method called partial least squares, PLS, where we're basically, again, assigning weights to codon frequencies that best explain the variation we see. And I should say that among all the things that we've inputted, codon frequencies themselves, even though not the codon adaptation index, but codon frequencies explain most of the variation we see. And this particular model is only based on codon frequencies in the gene. The most important statistic here is the cross-validation statistic, this R² CV, which is the ability of the model to predict left-out data. So what it means is when we leave, in this case, is when we leave 20% of the data out and we build a model with the remaining 80% of the data, how well does it predict that left-out data? In this case, it predicts it very well. We have a very good correlation coefficient for left-out data. So it's telling us that the model is not overfit. Another way we can tell that the model's not overfit is by basically randomizing the data, randomizing the expression values, and seeing how likely is it that this model is insignificant, that it's random, and it's very unlikely in this case, greater than 99% chance that this is a significant model. All right, so, and another way we've tested the model is we've made a few genes predicted to be good by the model or bad, and that's shown with the green triangles here. These weren't part of the modeling, but when we synthesized them according to what the model's predictions were, they were accurately predicted to be either good or bad. The bad gene in this case is a gene that actually was made with a high codon adaptation index, which the model predicted to be bad and turned out to be. We've also tested the results of the model with a number of different other proteins and compared to a high codon adaptation approach that's shown in this table up here. What we've seen, and we've seen this for, we've applied this model over many years now, or a couple of years now, we've seen basically this for well over hundreds of designs. But basically, we're seeing a consistent high expression for the model predicted codon frequencies. We see much more variability and often much worse expression when we use a high chi approach. So what we think we're doing is finding a sweet spot where we have more reliability and generally higher expression with this balance of codon usage. I don't have slides that get into the details or our speculation on the mechanistic basis for this. There's some speculation in the paper. I'll refer you to that for some of that. And we also have a review out recently in protein expression and purification, which also addresses some of these issues. But what we do see that's interesting is that our highly expressed genes in these sets are biased toward use of codons that are read by tRNAs that are predicted to retain their levels of amino acid charging under conditions of starvation or high overexpression. So under conditions where recharging of the tRNA can become limiting. And so we think that maybe what we're finding here is that we get better expression when we shift to a balance of tRNAs toward tRNAs that are able to handle the burden of overexpression. All right, so I said that we didn't see in the initial sets any correlation with RNA structure, but there's plenty of evidence in the literature that RNA structure is critical. So we wanted to add that into our modeling so to make our model more generic for new proteins, anything that could come down our production line. So one way we approach that is to build on the set of CUDLA, where this set built around the fluorescent protein, where an effective structure was seen. And the CUDLA data in this slide is shown in blue diamonds, so that's the data we're starting from. That's the correlation that they saw with RNA structure. We built on top of that set with a number of genes varied in codon usage and RNA structure. And interestingly, when we made a gene that was predicted to be good from the model based on the previous other two genes, we got an expression level that was 3 times higher than the best in the CUDLA randomized set. So we were able to not only expand the range of expression levels that we see by using the model-driven frequencies, but we also were able to improve a gene based on data from 2 completely unrelated genes. And not only that, we also see a lot of variation that can't be explained by RNA structure. A number of genes here that all have the same RNA structure strength, but vary widely in expression level. So we then took that data and we showed that you can build a model so that incorporates both RNA structure and codon usage into the modeling, and that's shown to the right here. And we get a very strong model that predicts both the original data as well as the added-on data for GFP. And indeed, we can incorporate all 3 of their gene sets together into one set and make a single regression that can explain all 3 simultaneously. And we get a very significant model here. I just saw a question that came in, and they asked whether we retested the CUDLA genes simultaneously with ours. And we did not do the entire set. We did a number of genes within their set, repeated their relative levels in our hands, and then normalized our new genes to those for this set. All right, so basically now this model, I mean, if you order a gene from DNA 2.0, this model is the basis for the optimization algorithm that we apply. So we're basically trying to match codon frequencies that are predicted to be good from these multiple gene sets that we have. So they're experimentally validated codon biases that we use rather than any guess from natural biases. And these, I'll refer to some of this information is in our PLOS One paper, the identification and levels of particular codons that were important. That's evolved over time as we gathered more data, but basically you'll see that that this doesn't correlate, in any obvious way to obviously the codon adaptation index I showed you that previously, but also just matching the codon bias of the host. It's a distinct shift. All right. so beyond E. coli, our main push right now is to expand the use of this technology to other host organisms of interest. And this slide represents some work that we did with Anton Gleiter at the Technical University at Graz, and Anton is an expert on the development of Pichia pastoris expression systems. For this study, we made 48 gene variants, varied similarly to the example I showed you earlier, varied in GC and purine versus pyrimidine content for each amino acid independently. And what they did is they took each of these genes and tested them behind 3 different promoters, so a variety of constitutive and induced promoters and a variety of different promoter strengths. And then they measured, instead of total protein production, they measured secreted cellulase activity. So they're looking at active secreted enzyme as the output. I will say, I'm going to take a quick break on this. We're getting a couple of questions. I'm going to try to handle most of those near the end. We'll try to grab a couple as we go through as well, but we'll try to come back to as many questions as possible at the end of the talk. All right, so what do we see with the Pichia set? So like the set in E. coli, we see a wide range of expression levels, over 2 orders of magnitude variation. And interestingly, we see a fairly high correlation between different promoters. So independent of whether they're constitutive or induced, these are all each promoter has completely different 5' UTR sequences. Growth conditions are different. They're expected to produce different levels of mRNA, but for the most part, a good gene is a good gene, and a bad gene is a bad gene when we look at them. There's some genes that fall off the correlation, so they might be interesting to follow up on, and we've got work going on with Anton to look at those. Another thing we see is that in our set, we see a lot of genes that outperform their previous best versions of these genes. So they've had multiple genes optimized by various companies for expression previously, and they got a range of expression levels. The dashed red lines here indicate the expression levels of their previous best, and just in this naive initial sampling, we were already getting a lot of genes that are better than their best guess at the outset. And like with the E. coli data, we can explain a lot of the variation by gene features in the PLS model, and this is the model shown that we get for their data shown here for one particular promoter, and we get similar models for each of the different promoters. Okay, so one key advantage, I alluded to this earlier in the talk, the key advantage of our approach is that we can get a lot of information from a relatively small number of genes, and that's particularly important for hard-to-assay proteins like human membrane proteins. In this case, this slide illustrates some work that we've done with Bob Stroud. Bob is a leading structural biochemist. He's got a strong interest in producing large amounts of usually human membrane proteins in yeast as well as some other hosts. In this particular project, we're expressing a human membrane protein in Saccharomyces cerevisiae, and what is assayed is the total protein in the membrane fraction. In this particular case, we're biasing a little differently from the previous example I showed. For each amino acid listed, we're biasing the codon bias either toward the bias already found in yeast, so increased bias, we're using the more frequent codons more, or we decrease the bias where we sort of flatten it. We're bringing up the lower frequency codons for that amino acid. And what we see again is a very wide variation of expression levels in the set. And traditional optimization schemes like matching the yeast bias or biasing toward high frequency codons gave us very poor results in this case. But we did get some really strong hits that were expressing at over a mg per liter in the membrane fraction, plenty of enough for them to take on to crystal structure studies. And the wild type gene, I should mention, in this case, gave no detectable expression. So they went from no and traditional designs giving low expression to getting something that they could take onto trials because of the sampling that we did with intervals. And I don't have the details here, but like with the E. coli sets and the Pichia sets, we can build a model that explains this variation very well. And we've done a lot more work with Bob Stroud, both in yeast and further work in mammalian cells as well. All right, so we now have many projects going on in a wide variety of hosts, and I'm just glossing over them here with the summary slide. But you have data in everything from gram-positive and negative bacteria, so positive in the case of Clostridium, negative in the case of E. coli. We've got multiple yeasts, Pichia, K. lactis. We also have some work going now in Candida, and I showed you some work in Saccharomyces. We also have data in higher eukaryotes, data in corn and also in mammalian cell lines. And we even have data in a mushroom, Agaricus pastoris, shown here, which is one of our best models and where we have a lot of data. Our big push these days is in mammalian cells. So we have a lot of effort going right now over this year to really build up our knowledge of the preferences of mammalian cells. And we hope by the end of the year to have more data in mammalian cells than we have in E. coli. So just in summary, our approach of experimental optimization using interlogs has shown to be very reliable for improvement of gene performance of any protein in any host that we've tested. And now we have early experimentally derived algorithms for many different hosts. And we've got many studies for many different hosts going currently, and we expect to have more data and stronger algorithms for these hosts over the next year or so, including multiple bacteria, as I mentioned, multiple types of plants, an array of fungi. Again, mammalian cells where we're pushing hardest, and a number of other systems. All right. And for further reading, there's a lot of information out there. You can refer to our PLOS One paper for some information on our E. coli work. We have a number of reviews out that discuss the field in general as well as our particular approach, including a recent review that should be out maybe even within the week in protein expression and purification. I think it actually is available online now in a pre-proofed form. And there's a number of outside discussion of independent discussion of our approach out there in the literature. I encourage you to look up. And with that, just thank some of our collaborators and sponsors. And I thank you for your attention. And I'll look over the questions list now and see if we can answer some of those. So there's some questions about the particular specifics about how we about our expression system, say for E. coli. In the case of E. coli, all of the work I showed was basically it's all plasmid-borne with strong promoters. Much of the work is done with T7 promoter vectors and T5 vectors, depending on whether it was done on site or outside. But standard expression, high expression conditions, LB, and we're just looking at the total accumulation of protein over a fixed amount of time, usually 4 hours with a strong promoter.
I'm going to actually read some of the questions for you,
Okay.
Mark. That way we'll capture it in the recording for people who want to watch this again and listen, or for those who weren't able to make it because of various time zones, etc.
Sure.
So one question we got was, did you try to include tRNA charging levels to your codon usage in mRNA model? So did you ever look at the tRNA charge?
We did not look specifically at the tRNA charging levels in our systems expressing different genes. It would be a great thing to look at, and we'd love to follow up on this, both on things like our mRNA levels. There might be another question in there on that because it comes up a bit. The specifics that, with regard to how the system is changing and interacting with the genes that are expressed at high or low levels, are things that we don't have significant data on, and it's what we're pursuing. So to some extent, you could say it doesn't matter. That may sound like heresy, but But basically, you could say the system is so complex, if we see a correlation, we're reducing the variables that specifically pertain to the gene design, and whatever the underlying mechanism is, whatever. And that's more academic than practical. But I think it's very important that we understand the mechanism to the extent that we can if we want to narrow down the variables and improve what we actually sample in these sets. So if we found that almost everything was due to mRNA instability, then we might want to focus on variables that might be more related to mRNA instability sites rather than codon usage. But and if we knew that there were specific tRNAs that were causing more problems than others, I mean, I think a lot, for example, of what we're seeing with regard to tRNA usage might not be strictly how much charged tRNA is available in some static sense in E. coli, but it's actually a matter of the response of E. coli to the loss of charging levels. It's well known, it's been well established in fermentation literature that overexpression of proteins can lead to a astringent-like response where you actually start degrading your translation machinery to a large extent. And these secondary effects of overexpression may be what limits your yields, and they may be differentially triggered by genes using different codon usage. So if we overuse certain codons that stress the tRNA population, that increase, the resulting increase in uncharged tRNA may lead to a response that limits your expression. So it may be that we're finding a balance that the cell can accommodate the most. Because really what we're trying to do here is pretty unnatural. We're asking cells to accommodate something that, the closest analogous situation in nature is maybe a viral infection. And cells have evolved ways to really shut down foreign protein production, and we're trying to get them to do it for us. So perhaps optimization of genes is more a problem of avoiding the natural preventative mechanisms of the cell to get them to do what we want.
All right. Another question is, does each project tend to need its own model?
So one thing that we'll learn as we do this more, and we don't have, I would say, enough data except for maybe E. coli to say this generally, but we need to know to what extent the models are basically protein dependent. And what we do see in E. coli is that there's a protein dependence in that the amino acid content of the protein seems to have a strong influence on which codons are explaining the variation. So a protein, say, that is highly rich in serine, like our antibody gene is much more dependent on serine codon bias than the polymerase gene that we studied. We can rationalize that in that explanation, or in that the idea that we're stressing, if you have more of a particular amino acid, you're stressing that part of the translation machinery more, and so maybe that's why those codon biases are more important. They become rate limiting. So for E. coli, it turns out different proteins with very skewed and different amino acid content have different models that are more dependent on the amino acids that are more prevalent in those proteins. So you might, to get a model that's generic for a particular host, you might need to sample data from multiple proteins. We also, if our output level or if our performance measurement is different in different cases, so if we're looking at, say, soluble versus total, then yes, we might get a different model there too. We may have a model that actually points to some balance that maybe even slows translation to maximize some balance of folding and output. We just don't have enough data to say that comes into play in a significant number of cases. The case that we, in Pichia, where we looked at total secreted enzyme activity, we saw that our expression levels, our secreted expression levels, correlated very highly with total protein. So the active protein correlated very highly with total protein limitation of the poor genes was mostly a limitation of production, not of folding or secretion in that case.
Okay, another question is, how are you codon optimizing for HEK293 at present for ordered genes? Is there more than one option since you have work in progress?
That is a very rapidly evolving model. So we had a one gene model, a single chain antibody expressed in HEK cells that we generated a couple years back, and for various reasons, we want to expand on that. We think it's a very solid model, but we want to add a number of genes on top of that one to build a real reliable model. And we have data pouring in. We really have some really interesting early modeling studies in mammalian cells for multiple different targets going on at the same time right now. We're hoping over the next couple of months to have a very nice, very broad data set for multiple targets.
Okay, two other questions that are related, so I'll tell you both of them, then you can answer them together. Can optimization also optimize solubility of the protein E. coli? And can you optimize toxic genes for better yield in E. coli?
Yeah, that's a tough one. So I would guess that the variables that we're varying are not ideal. So just strict codon usage biases are not the ideal variables to sample the effects on toxicity. We could use the same approach. We just might want to redefine what variables we look at. For solubility in particular, say we might want to look at intentionally, we want to look at a distribution of rare codons or rare codon clusters or things thought or determined by our earlier data to pause translation. Maybe strategic pauses could affect solubility. This is one of those cases where, for both toxicity and solubility, there you really expect that whatever solutions you come up with are not going to be generic. They're going to be very protein specific. So I think the idea of experimentally sampling and basically using more of an iterative engineering approach that we're talking about here is ideal for those situations, but it's going to be a different set of variables, I think, than those that control total expression yield. So we have to, and again, the models that we get, depending on what we learn, we may learn some general principles that we could apply to multiple proteins, but we may end up with protein-specific solutions in each case. For toxicity, it's more complicated. Now we're talking about, in most cases, we're talking about the properties of the protein being incompatible with the host, and those are cases where we might want some balance between expression level. We might want to lower expression level to some optimal level where you're producing the most without killing the cell. And I think that there's some we can do on the protein coding sequence level to improve that. Controlled promoters might be a better option for that if knocking down expression is the issue. So yeah, and again, those are going to be case by case, protein by protein, and host by host solutions.
Okay, another good question is, why do you think that chi or 5' mRNA structure were not affecting the gene expression of both genes?
Okay, so the case of structure, I'll reiterate that our data is pretty consistent with what's been seen in the literature, that only really strong structure gives you a strong effect on expression level, strong structure near the ribosome binding site. And we didn't see that in our gene set in our first sampling in those two for those two proteins, and that's clearly we've made all possible structure, all possible sequences for those genes just to sample the range of structure strengths possible for those genes, and they're really pretty weak just because of the amino acid sequence of those proteins. The amino acid sequence determines 2/3 of the nucleotide sequence, and some genes, just because of their amino acid terminal sequence, aren't prone to form strong structure. and that's the case for our two test genes. We can make them strong if we, I mean, there are some stronger variants of those sequences, and we've shown that those will inhibit expression when they're strong enough. But in our sampling set that we published in PLOS One, we just didn't have very many genes that were of strong structure enough to see a significant effect. So we've seen that when we've looked at other genes that are more where we can put stronger structure in more readily, like the fluorescent protein example that we described. And we're fully convinced from that data and a lot of other in-house data that strong structure can be deleterious. There's a lot. We hope to publish on this soon, but we are spending a lot of time here on trying to understand the effects of 5' coding and structure, because we know that just from our own internal data, and this is unpublished stuff, but there are a lot of effects that we can't explain by local RNA structure, but are clearly confined to the 5' end. And a lot of very interesting stuff going on there. It's not a simple structure strength as predicted by these folding programs problem. It may be structure that's not well predicted. It may be tertiary structure in the RNA that's hard to predict from these folding programs. There's a lot of complexity there that we need to delve into to fully explain the effects on the 5' end. But what we do know is that local 5' end effects don't explain most of the variation we see for most hosts. If the structure is strong enough, yes, it'll trump the effects of codon usage throughout the gene. We can inhibit initiation. But once we remove structure, we still have a lot of variation that is critical to manage for gene optimization.
Let's do one last question, which is, can you optimize a gene for expression in an organism for which you do not currently have a model, or do you need to develop a model first?
So what we do, our default is similar to what other people do if we don't have a model at all. So we'll usually we use the organism bias, the natural gene bias of the organism as a starting point. So we can always do the more traditional optimizations. We'll still manage RNA structure, and we'll still avoid rare codons and match host bias as a default if we don't have experimental data. But I will say that in a number of cases where we've actually gathered experimental data, we know that those approaches are pretty poor. But it's the best starting point if you have nothing, I would say, until we learn more. But we found organisms where the optimal bias is considerably different from say the host bias, and the host bias can really underperform in some cases.
Great. Well, I'd like to thank everybody for joining us today on our webinar. I'd like to thank Dr. Welch for his wonderful information. Lastly, I'd just like to invite everybody to join us for our next webinar, which is Thursday, April 5th, and we'll cover design of protein variant libraries to examine sequence diversity.